speakers and welcome all of our participants from Canada and from across the US. Welcome. My name is Amy Liebman. I'm the Director of Environmental and Occupational Health at MCN. And I just want to check in with Martha and Kate and our great team that's working sort of the wizards behind the curtain here. Are we ready to start recording, and are we ready to start with Facebook Live?
Yep, we're all set up on Facebook Live. I think it just started streaming and it looks like we're already recording.
All right, now that's live.
Excellent, excellent. Okay, I'm gonna go ahead and introduce our speakers today. We have a great question and answer discussion that we have planned for you. Is COVID-19 airborne? It's a Q&A with Dr. Chad Roy and Dr. Ed Zuroweste. Let me start by introducing Chad Roy, and we are really, first of all, thank you for taking the time out of your day to chat with our network and our stakeholders. We're really excited to have you with us. Dr. Roy is a microbiologist and director of infectious disease, aerobiology, and biodefense research programs at Tulane University's National Primate Center. He's focused his career on respiratory health and aerobiology of airborne infectious diseases, specifically on gaining a better understanding of aerosol infection in the context of the development and application of preclinical disease models. The majority of efforts are directed in the use of non-human primate for this purpose. And we're super excited because he's currently engaged in some of the latest COVID-19 research, including being the principal investigator of NIH grant to evaluate the nation's most promising vaccines and treatments to address COVID-19. So welcome, Chad Roy. We're really excited to have you with us. And then it's always my pleasure to introduce Dr. Ed Zuroweste, who has been a frontline family physician for 30 years. He cared for many farm workers and their families in Chambersburg, Pennsylvania, and he is the founding medical director of the Migrant Clinicians Network. And he's also many other things in addition to the founding director of MCN's being medical director. He is a tuberculosis medical consultant for the Pennsylvania Department of Health. And he's also served as a special medical consultant for the global influenza program of the World Health Organization, where he helped limited resource hospitals and clinics during the H1N1 pandemic, and was part of the WHO Ebola response team, where he trained physicians and nurses to prepare for work in Ebola treatment centers. So with that introduction, we'll give a warm virtual welcome to both Chad and Ed, and I'm gonna hand it over to Ed, and to Chad, and I'm actually gonna turn off my video while Chad's speaking, and then we'll go ahead and start with the Q&A after that. So I'll hand it over to you, Chad.
Thank you so much, Amy, and it's such a pleasure to be here today. I'm humbled to be with Ed when how accomplished he is on the clinical side of things. Some of the things that we're doing now hopefully will be informative to the current situation, the pandemic that we're in right now. I've been very fortunate to be involved at an early stage with it in terms of experimental medicine and experimentation in general on this new virus that back in September of last year, remember, we didn't know what it was. We didn't even know about it until we heard about it as a unexplained pneumonia in Hubei province in China, where it was first went on ProMed, and then that's when everything started to try to characterize it. The genome was soon worked out by colleagues in Australia, and so on. So tracking it now and all the wonderful work that's being done and so many people that are involved in this, I feel fortunate to be a part of it on the experimental side in 2 specific areas. So over the next couple of minutes, I'll just briefly describe what we're involved in, what we found thus far, which is of interest in terms of characterization of this virus in aerosol, and what we know about it, the physical and biological constituents of it, and how that could possibly impact how we protect ourselves against it, the transmissibility of it, and ultimately the pathophysiology of it when we're testing what we're referring to as medical countermeasures or vaccines and therapeutics. Those have to be tested for safety and efficacy, usually using animal models, many times non-human primates. So the characterization of that in the laboratory is absolutely critical to inform those processes, so there ultimately can be effective medications to get out. But it's very helpful to talk about the characterization part of it first to inform individuals like Ed, right? That's on the clinical side of things, and need to have a deep understanding and appreciation of this virus and how it moves from infected individual to naive individual, and how we can best protect ourselves. So over the last several weeks, and I say weeks because everything is moving so quickly, in my laboratories, we've worked on characterizing what happens to this virus when it's in the air. And so that, what Amy mentioned, the aerobiology of it, so that the ecology of this virus when it's in aerosol. And many of you have heard on the news that it only travels in large droplets, and that's why we're doing the 6 feet apart. That's some of the basis of that, based on probable transmission pathways. So a lot of that has been based on very basic information on how this virus is infective. But these types of studies that I'll describe just very briefly tells us a little bit about the smaller fraction of those aerosols, and those aerosols that can move through the air over longer distances, and whether or not this virus has a propensity to remain infective over longer periods of time in the aerosol form. So it's a very kind of dark little corner of science, and not a lot of people engage in on understanding infectious aerobiology of different high consequence pathogens like this. So the first thing we did is we reached out to colleagues at the National Institutes of Health, other universities. University of Pittsburgh was involved, and the U.S. Army. The U.S. Army Medical Research Institute of Infectious Diseases, USAMRIID, that has a great capacity to do these types of studies and study and characterize aerosols. And then in Tulane University. And so for disclosure, I was with the Army for a number of years before I came to Tulane. So I have colleagues there with similar labs. And what we did is we took the virus from a Seattle strain that we received. We grew it up, and then we generated it using an aerosol generator into a chamber. And we did that with the SARS-CoV-2 virus that is a current virus in this pandemic. We also did it in similar experiments with SARS. You know, some of you may remember SARS-1 back in 2002, 2003, causing an outbreak. And MERS, the Middle Eastern Respiratory Syndrome virus. All of these are emergent coronaviruses over the last 25 years. And through a variety of machinations and details that I'm going to spare everybody from, we compared the aerosol efficiency in short-term dynamic environments. So the residence time of these aerosols were about 30 seconds to 1 minute, just to get a brief knowledge of infectivity and residual infectivity in aerosol. And so the synthetic aerosols that we were generating in the laboratory under controlled conditions and trying to control everything around that and doing it in a safe manner as well, which is a feat in itself. We determined that the SARS-CoV-2 virus was more efficient in aerosol than those other two emergent coronaviruses. And the infective fraction of the virus was much higher than the other two viruses. So it had what we would think in kind of a remarkable in the words of that, it's suggestive that this is more efficiently transmitted by aerosol than, say, SARS or MERS in the context of those three emergent coronaviruses. Now, that's not speaking to flu, for instance, or other things. I should also mention that when we were doing these experiments, we were using a particle size that would be consistent with a respirable aerosol. And remember, we were doing this in a manner to where we were generating it in a chamber in a very controlled environment, so we could study it. We weren't exposing any animal to it or anything like that. We're just studying the aerosol. And so when I say a 2-micron particle, what does that mean? So, you've heard larger particles will drop out of the air in 6 feet and that sort of thing. This would be smaller than that, but it would be a liquid aerosol that kind of inside that aerosol, that's where the virus would reside. So these viral particles are very small. Everything's small, but these are very small. These are about 110 nanometers in size. So in a 2-micron aerosol, you could have 3 or 4 or even 10 viral particles residing in a single aerosol. And why that's important is that for instance, our protective mechanisms of an N95 mask protect against those types of particles. They are above the threshold in which an N95 will protect, which is great news for everybody. And we decided to use that size particle to study the efficiency and infectivity of these particles, and we did that across all 3 of those viruses. So after we did that and determined that SARS-CoV-2 had a higher efficiency than those other 2, then we wanted to understand how long does that happen. And so we just focused on SARS-CoV-2, and we suspended that aerosol over many, many minutes. And again, there's a lot of methods associated with doing that to make aerosol suspensions that will not settle out. We use some interesting pieces of equipment to do that, again, in the same laboratory, but just focusing on SARS-CoV-2 to understand how long does it take in those 2-micron aerosols for the infective fraction to disappear, basically, to biologically decay in aerosol. So we have some understanding of that. Now, this work was done on the heels of a paper that was reported in New England Journal of Medicine that was done by an NIH laboratory that showed those types of aerosols in suspensions at about the same particle size will survive about 3 hours in aerosol. And that wasn't because this is a colleague that did this work. We had conversations about it. This wasn't because they had determined at 3 hours the infectivity is gone. It's just they had done that experiment just for 3 hours. So 3 hours wasn't a magical number to where the infectivity would disappear. So we felt it obvious to, extend those time periods to see when, in fact, the aerosol infectivity starts to drop off, kind of like a biological half-life in aerosol. And so we extended those studies all the way out to 16 hours. And so we did this. It was actually done overnight in the laboratory. We had people sleep there to watch the experiment. It was that important. And we sampled along the way to see at 10 minutes, 30 minutes, 1 hour, 2 hours, et cetera, what kind of curve we were looking at in terms of the biological decay of this virus in aerosol. What was really remarkable about that experiment is that at 16 hours, there was still an infective fraction of a virus in those aerosols. And that was remarkable in that I've worked with a number of viruses through my career in doing kind of the same types of experiments. And in influenza, for instance, we don't see that. We see it decay after a couple of hours in aerosol. With others, other viruses, it may take a little bit longer, but we haven't found one that lasted out to 16 hours like this SARS-CoV-2. And remember, this is in a laboratory controlled environment. The translatability of these types of results is not direct, meaning, oh, well, it must last 16 hours out in the environment. That's not this is only suggestive of under perfect conditions for that virus, completely in the dark, a temperature and humidity that would be consistent with survival, that sort of thing. It's a piece of the puzzle. It's not the whole puzzle. But it is remarkable, and it is very worthy information to get out there to policymakers, to folks that have their eyes on all of the data, so they can say, oh boy, this has an unusual persistence in aerosol under these conditions. Maybe we need to look at it further. Now, since I've done those experiments, and we put all those experiments in a manuscript and put it on what's called a preprint server, basically waiting for it to be published, because it takes a long time in science for things to get published. For those of you that don't publish in science, it can take years to get into a journal of any repute to get that information out there. We thought this was important enough to put on the preprint server. medRxives is the name of the preprint server, and it's kind of like a purgatory for scientific information waiting to get published, because it's not peer reviewed, which is required for publication. And this has since undergone peer review, and will be published very soon. I just can't say which journal because it's not accepted yet, but it will be coming to a journal, near you soon. And it goes through all the details of the experimental conditions and things like that we used for this. And so in my experience of over 24 years of working in infectious disease aerobiology, those types of basic experiments, and again, this was preliminary data, so we're doing it over and over in the laboratory as good scientists should do, and we've been taught to do. That may change a little bit, but as a first blink on what we're dealing with, and it does answer a lot of questions in our mind about transmissibility, the relatively high R naught that has been, I guess, just published in the last day or so, that has moved it almost to a 6 as an R naught. It answers some questions, and it makes it very plausible that aerosol and airborne transmission is a predominant, it's not a replacement, but it's a predominant exposure pathway for infection. So that, when we're looking at the collective of how people are exposed to this, infected by this virus, almost think about it as an extra highway being added to one or two highways that can lead to infection. And it was only through this type of data, and keep in mind there's other laboratories that are involved with this that are independently confirming this. It makes us really, appreciate the possibility and plausibility of aerosol and airborne infection as a possible route of infection. So that's a brief summary. Hopefully I didn't take too much time on the characterization of this as an aerosol. Those studies, like I mentioned earlier, are going to inform medical countermeasures, so vaccines and therapies for the infected in animal models. That's mainly what we do at the National Primate Research Center, and a characterized model of disease, so we can make sure that anything that is going to go into a human being is safe and effective before it goes into a human being, which is absolutely critical for this. So we're doing that hard work now. Now that we have a good characterization of that model, we understand a little bit more about transmissibility as it pertains to the aerosol route and impacting the respiratory system. I don't have any details on specific vaccines. I know a lot of you have heard in the media that they have this vaccine or that vaccine and it's been in humans. The one thing that I can say to please keep in mind, and Ed will probably have some wise words about this, is that when you do hear in the media that this has been in humans and they're going to start manufacturing it 6 months from now, that is in the context of safety and the initial immune response associated with that vaccine administration. It has nothing to do with the efficacy of that vaccine. And because they have to do that in people that are infected or going to be infected. And they're not doing that yet. The safety studies are ongoing in the clinic, but that doesn't mean that it's necessarily effective. It may be effective, but that hasn't been determined yet. And many of the organizations that are putting these vaccines together, in conjunction with like WHO and all the NGOs that work with, which has been incredible, this public-private partnership in trying to get these vaccines as soon as they can, they concurrently manufacture these vaccines with the anticipation that they will be effective in human beings. And I should mention also, because I'm a little involved with this, is that they concurrently run animal studies to see the safety and efficacy of those vaccines as well. So there's a number of different shots on goal that can bring us a vaccine. But, please don't get the idea that by the December, we're going to have a vaccine that's going to be actually licensed by our Food and Drug Administration for human use. There's still the burden of safety and efficacy in human beings that they're not giving a pass to anybody for this infectious agent or any other. They have to do their job. And so anyway, that I just want to kind of say that as a disclaimer, because it's being on this side, and I'm sure Ed feels the same. It's a little frustrating when you hear the news that, hey, they're putting it in humans and they're manufacturing it. It's just natural to think, well, why aren't they just producing it next month? It's that easy, and it's not.
Well, thank you so much for giving us that really amazing overview and sharing the work that you're doing. We have a lot of participants, and so welcome everyone. And we also have a lot of questions. We had a lot of questions during the registration process as well. So I just want to start off our Q&A before I hand it over to Ed for a moment, and to let you know that we're going to do our best to respond to all of your questions. And if we can't do it within the hour that we have with you today, we'll work on getting some answers, and know that we're doing our best just to get this preliminary information out to you, and some ways for you to absorb it. And for those of you on the front lines, what does this all mean? So with that, we'll turn it over to Ed for a little bit, because I'm really curious to ask Ed, just in hearing Chad speak as our frontline clinician, and some of the questions that we've heard from our clinicians and frontline workers across the country. What are you thinking right now?
Yeah, that's great, Amy. Thanks a lot. And I just want to remind everybody, today we're focusing our discussion on the airborne potential of COVID. That's all we're going to talk about today. So other questions about COVID and how to protect yourselves and your patients. That's what we're going to focus on, because that's a big topic right there. And as Amy stated at the very beginning, we want to make sure that we're providing to all of you the most up-to-date science as we have it right now, as of May 13th. So May 28th, some of what we say today may change. So I always like to preface anything we talk about, especially with this particular virus, that everything is moving so quickly. We just want to make sure that this is the best we know today. And what we're going to try to share with you is the pure science. And we're not here to set policies or procedures for you and your clinics. We're here to give you the science, and then it's really up to you to take that science and decide what you want to do with that. First of all, I really want to thank Chad and his colleagues, because it's really this bench science that is so important for us to know, so that we know what to do with that information. Without this sort of science, basic science, we really don't know how this virus is going to act. And I think all of you in the very beginning, we all felt that, in the very beginning, they said it wasn't airborne at all. And then as time's gone on, with a New England Journal of Medicine article that came out and said it could be in the air for 3 hours, then people start saying, well, maybe it is a little bit more airborne. And I think what you've heard Chad say today, it really does seem to be a significant part of this virus. So what I want to do over the next few minutes, and I'm just going to take a few minutes here, is to take that bench research, because it's always the question is, you have bench research, but then how does that translate into my world, into your world? How does it translate when we're actually dealing with patients? And when you have that virus not in that chamber that Chad has at his highfalutin, research center, but you have it now in our clinics, what does that virus do in our clinics? And so what I wanted to do was share with you, and Amy, if you can put up the slides now.
Okay, coming your way.
I want to share with you what we're seeing now in epidemiological studies that really help solidify exactly what Chad has just been saying. And that is that it appears this virus is much more airborne than we thought. And this, I think, is a great picture to show if somebody sneezes, that's what comes out. And as Chad said, some of those big particles, some of those bigger white particles are just going to fall to the floor, and they're not going to go farther than 6 feet. But then if you look at the rest of the atmosphere there, it looks like a summer night there. A lot of those very tiny particles are going to, as Chad's research has shown, may stay viable in the air for 16 hours. And if they do, they're not going to stay in one spot, and they're not going to stay within 6 feet. So if you go to the next slide, Amy. So there was been a great paper that was been put out by Dr. Aaron Bromwich, who's at Dartmouth, and he had compiled many studies, and we've got that as a handout for all of you. And I think it's worth the read. It's pretty long, but it's worth the read. And so this was one study that was done by epidemiologists. And if you look at this, this is a restaurant, and the people at table A were there for about an hour, hour and a half. And A1 was an asymptomatic COVID positive person. If you look to your right on that screen, that's where the air conditioning was. And if you can see the arrows, the air is going from right to left, from table C to table A to table B. So during that hour and a half at table A, 4 other people of that 10-party dinner party got infected with COVID-A and presented within a week. 3/4 or 3 out of 4 of table B got infected, and even 2 out of the 7 people in table C got infected within a week with COVID. So this shows that probably a significant part of this was the airborne component. I think it would be hard to describe that somebody from table A handed a salt shaker to table B and they passed it around, right? Some people at table A may have been sharing some things, and it might not have been air, but I think this is a good example of that probably airborne was a significant portion of this transmission. Then if we go to the next one, Amy, the next one was just published yesterday. This is how current we are at MCN. We want to give you the information as soon as it comes out. And this came out electronically from the CDC. This is an MMWR that hasn't been published yet, but it came out electronically yesterday. And it shows this was an epidemiological study in Washington State at choir practice. It was a choir practice for 2.5 hours. So they're all singing very enthusiastically, I'm sure, in their church. And there was one symptomatic person, and that's the person in red. And that person infected 32 confirmed and 20 probable cases of COVID. And of that, the mean age of these people were 69. So this was an older group. So the attack rate was either somewhere between 53.3% to 86.7%. 3 of these people ended up in the hospital, and 2 of these individuals end up unfortunately dying from this COVID, from this 2.5 hour choir practice. Again, there may have been some handshaking and hugging and that sort of thing, but probably a significant part of this infection rate was airborne. All right, so, and if you look at, there are several other studies that Aaron has put in this paper to show the very much potential of this airborne. So what Chad has told us that is pretty well defined now in the laboratory is now acting itself out in real life. So that we now have to say that this virus has significant capability of transferring air through the airborne route, and not just 6 feet. Obviously, it depends on where you are, what kind of room you're in, what the air transfer is, and how long you're in that room, whenever we're looking at things like this. So then the question I would have is, so how am I going to protect myself? How am I going to protect my patients? What am I going to do in the clinic? So that's where masks come in. So Amy, if you can put up the next slide. So this is Eva Galvez, who's a family physician, and she's on the board of directors of the Migrant Clinicians Network. And you see her in full PPE. She's got a N95 mask on, she has goggles, she has a shield, she has a gown and gloves, because she's going to test this child for COVID. And so that may produce some aerosol, and so she's protecting herself. And the reason we say N95, the name means that at least 95% of these tiny particles that Chad was just talking about are not going to permeate that mask. And remember, the N95s have to be fitted. They have to be fit tested to make sure they form a seal on your face. And if they are fit tested, you have at least a 95%, if not greater, assurance that that viral particles aren't going to pass through there. All right, so that's the N95. There's 3 basic kind of masks. The next one, Amy, if you'll show, is our own Dr. Medeiros, who's now the Chief Medical Officer of the Migrant Clinicians Network, and he has a surgical mask. And so surgical mask, and I'm just going to read this to you, and this is from the CDC, does not reliably protect wearers from inhaling tiny aerosols, okay, because it's not fitted to your face, and he's got a beard, and you look at my face, you put a surgical mask on, it's very possible that those tiny particles are going to sneak around there. So it's not a reliable protection. for these tiny particles. And then you have the homemade masks that are just cloth masks that people can make at home, and they're even less protective than this surgical mask. So where are we in May 13th with masks in the country? Well, it was interesting in January and then again in February, the CDC recommendation came out that N95 mask and full PPE were to be used, and I'm quoting now, when evaluating or treating COVID-19 patients or suspects who might have COVID-19. N95s were recommended. However, on March 10th, the CDC downgraded those recommendations to only need the N95 and full PPE when dealing with COVID patients during procedures that might cause aerosolization. So they downgraded it, and that downgrade, I have to say, was not based on science, because there was a lot of people in science who thought that was a bad idea. And apparently it was because of the lack of N95s, a lot of pressure from hospitals because they said we cannot meet these standards, and a lot of pressure from Congress, again, and the administration. So I'm just saying that we always try to agree with our CDC and our WHO, but sometimes we have to look at science. And I think that right now, the recommendations that were in January and February from the CDC were very accurate, and that now N95 masks are the only thing that really are going to protect you if you're in with a COVID patient. So I'm going to stop there, I think, Amy, so that we have time for questions. And I just wanted to get that little bit of background so that we can go on to questions now.
Great. Well, thank you so much. Well, both of you have given us a lot of food for thought. And I just wanted to let you both know that we have a lot of questions. We got a lot of questions when people registered, and we have a lot of questions on the list. And so I'm wondering, Chad and Ed, if you'd like me to just start off with some of the questions that came in through registration first, and then we'll try to turn our attention to some of the questions that are coming through the chat and the Q&A. How does that sound? Okay, so, and again, thank you so much for submitting your questions. The risk of transmission outdoors would appear to be extremely unlikely given the rapid dilution of the respiratory particles and sunlight. Can you discuss that?
Yeah, absolutely. It's a great segue into contextualizing some of the research that I talked about earlier. And so, first of all, there's data now, as Ed said, it's happening every day, right? We get data in every day from colleagues that had the same sort of setup that I had, but they expose the virus and those viral aerosols to UV light that would be consistent with sunlight. And that magically went from 16 hours down to a minute and a half. So highly susceptible to light at those particle sizes that I was speaking of. So it's all a matter of context. That's why I was stressing and emphasizing that it's only piece of the puzzle, kind of worst case scenario here. And then as this data informs, you get this collective of, okay, what is the risk here in terms of transmission? So all those data points are important. One doesn't outweigh the other, but it's great when we have that context. So let's think about outside, working outside, whatever it may be, jogging, that kind of thing. First of all, you're in millions and millions and millions of cubic meters of air. So the dilution effect on these particles that we're talking about, which I was working with highly concentrated aerosol particles, is massive. It's unbelievably massive. It's so big that it makes the probability of actual exposure infinitesimally small. So, and this is, kind of outside in a park small. So that weighs into the overall probability of you being exposed to, remember, it takes probably more than one viral particle to induce infection. There's a multiplicity of viral particles that you're exposed to in order to become infected. So it's not like the one hit theory. You get one viral particle in you and you're gonna get sick. So considering that there's a response to that sort of dose and that incredible dilution, and then what we know now about sunlight, and different things will come out in terms of humidity and temperatures and things like that, you can see how the relative risk would be reduced over time. So say wearing a mask outside, it's, you know, it's not something that I would consider in trying to assess my risk to this virus, unless I was at a rock concert where I was shoulder to shoulder with folks and everybody's yelling and screaming, generating bioaerosols that I could potentially be exposed to. So there's a dose of, you know, this translatability of some of the experiments that I talked about and those results that Ed did a good job of saying, look, you know, we need to assess the risk per our situation and scenario and the probability of exposure. So I think, you know, everybody does it. I do it. It's innate with a lot of us to make like this binary choice in our mind. Like either you're going to be exposed or you're not going to be exposed if I do or don't wear a mask. And the fidelity in that decision-making process is so critical to assess that risk based on what we know. And that's why with this, it's especially poignant that everybody in science and clinical and everybody is rushing in urgency to get as much information as possible to make those decisions. Just like Ed mentioned, you know, May 28th, it may be a different situation based on what's coming out, what we know, how this is developing in the human population. So the leadership in public health and otherwise are making the best choices with the data that's in front of them. And we have to assume, which I think is a pretty safe assumption, that everybody's trying their best to protect each other in this situation. So all knowledge is good, and in that regard, it makes it a lot easier when you have experimental data, for instance, to back it up or to have perspective on the whole thing. So I would say in terms of the outside, you know, I think it depends on the situation and considering all those other, you know, environmental factors and that sort of thing.
Great, thank you so much. Again, thanks to everyone for joining us right now. I just want to make a couple announcements because we have a ton of questions coming in. First of all, we have put together a packet that has a lot of different resources for you about respirators, about the two studies that Ed mentioned, the preliminary work that Chad has out there in purgatory, as he called it. We have links to all that, and that will be coming to you after we're done here. And then I did want to move on to our next question. And also, I'm seeing a lot of great comments and some resource provision, and some of you guys are just sending them to the panelists. So if you have a resource that you want to share, go ahead and send it to the attendees. Our next question comes from an outreach worker who's part of the Migrant Clinicians Network. And he has he's an outreach worker with a pre-existing lung disease. What is the best practice to protect myself when I'm out in labor camps or confined workspaces, such as at the health center?
You know, I'll field that one, Chad. You can add in. I think if you've got a pre-existing condition and you're out at a labor camp, just as Chad said, if you're interacting with farm workers outside, I would have the wind at your back. You know, this is something we learned from TB a long time ago. You know, tuberculosis is an airborne disease, and I've worked with TB for over 30 years, and whenever I've worked anywhere, if I can be outside interviewing people with the wind at my back going towards them when I'm talking to them, I feel much safer. So he should do that. It wouldn't be a bad idea for him to always wear at least a surgical mask. If he has to go inside the camp, like the housing of the camps where it's going to be a closed space where there's several people there, then I think he should wear an N95, especially with his pre-existing conditions. If he's going to be in a closed space with potential COVID patients, then he should wear an N95. Otherwise, I think a surgical mask would be fine if he's just out and about, or even in his clinic, and he's not really in a closed space with COVID patients.
Great, thank you. So our next question that we have, someone's asking about the usefulness of cloth face coverings. Are they helpful at all? Who wants to answer that?
Yeah, I'll start on that. Supposedly, they're not very helpful at all, except any barrier, and Chad, you can help me with this. Any barrier, if I happen to have COVID myself, and I'm talking, and I'm going to expel it, if I have anything covering my nose and mouth, I would assume that at least part of that, especially the big droplets, will be impeded by that cloth mask. It may not totally, some of them may permeate that, but it seems like even if they do permeate it, it won't be spread as much. So the cloth mask, I think at least will help from the person who's wearing it not to spread it as much. What do you think about that, Chad?
Yeah, absolutely. And so it would be consistent with the protection that one gets when wearing a surgical mask. And that's why we call it surgical masks, because they wear them in surgery. So the doctors and the nurses don't infect the compromised patient on the table. So when they speak or yell or whatever they do, and this is a truism, we all express bioaerosols, even through soft speaking. There's a certain number of bioaerosols that come out of your mouth. Thankfully, most of them don't have viruses in them. It's just a liquid bioaerosol with not much in the biological load of that aerosol. And when somebody wears a face covering like a cotton or polyblend, whatever it may be, over their mouth, that impedes the particles from going forward, especially the big ones. And when I say big ones, over 5 to 10 microns, that could be caught in the matrix of that. If there's multiple layers, for instance, of that, of course, it's not very easy to breathe through that, but that'll increase the ability of that to just physically catch these particles. We also need to remember that when we inhale, when surgical masks are worn or cloth masks, for instance, the pressures in the air work just like water. And so it's going to take the lowest pressure possible in your inspiration. And so that's why it's very difficult to say that, say, a surgical mask or even a cloth mask protects, especially against the small stuff, because it'll move right in the cracks of where it doesn't form fit to your nose, as you guys probably saw from what Ed had put up. Those N95 respirators, that's why they're called respirators. They're form-fitting to the face, and their matrix, what they're made of, will stop particles of 0.3 microns and above. So it's really small, 0.3 micron and above, of non-oily aerosols. That's how they're tested, and that's why they're certified by NIOSH to do just that. When you have a surgical mask or a cloth mask, that's not necessarily the case. Even penetration through the cloth mask or in those little crevices around, because of the velocities in your inhalation, in your inspiration, is very, very high, even though it's a microsecond that those happen. And so for a very small particle, like a 2 micron particle, for instance, that can be pulled in and you wouldn't even know it, because these are invisible. I mean, they're so tiny as to not be seen by the human eye. So that's the distinction between those masks and how they protect. Now, in the case of somebody that does have this disease, absolutely. Surgical mask, cloth mask, they do an incredible job of protecting others from transmission, but not vice versa necessarily. And it's just because we haven't quantified that, right? Those cloth masks haven't been tested. Surgical masks, to some extent, haven't been tested to see what they protect, but we know how they fit on the face, and the matrix that they're made of, that they're less than ideal for protection.
Great, thank you. So lots of questions coming in. A lot of folks are addressing questions and resources just to the panelists. So I'd like to encourage you to sort of, if you have great resources to share them with everyone. And then I mentioned that there was a lot of my colleagues in the occupational health and safety world that are on here, and they're sharing some great ideas, particularly about various kinds of respirators. And so I think what probably will be the best thing to do with all the information is I'm going to throw it back at you, follow up with you, and MCN is going to put together a blog with some of the resources and recommendations regarding some of the respirators that you're talking about here, and some of the tools for that. I am going to go on to the next question. And also, a couple things. Yes, this is recorded. Yes, you'll get a copy. It's also a CME and a CE event. So we ask when the evaluation comes up for you to fill out the evaluation. There's going to be a resource packet that's sent to you. And then we have a wonderful writer and a blog at MCN. And Claire Huckensada will be putting together a blog that kind of describes the discussion that took place, responds to some of the questions that we couldn't get to. But in the meantime, I'm going to look at some of our other questions here. And I want to see what you all recommend. Some of the questions are about what do you recommend for farm workers that need to be transported to and from work in a van or a bus? Should they wear masks? And what about the staff who's driving these workers? So there was another question about bus drivers. So maybe we could answer both those questions.
All right, well, I'll start with that. Again, we're not here to set policy and procedures, all right? We're here to tell you what the science is. So the science tells us that even if you're asymptomatic, and we didn't talk about this, but we know that a significant number of people never become symptomatic, maybe 40 to 50% of people infected. And even those that are become symptomatic, the first 2, 3, 4 days, they're not symptomatic and they can show. So we don't know. Obviously, if anybody's sick, they shouldn't be getting on a bus or a van. If they have a fever, they don't feel well, they have any symptoms at all of COVID, they shouldn't be getting on the van. If they have to get in a van or a bus, they should be separated as much as possible. And this time of year, I would open up all the windows of that bus, and I think they all should wear masks, and the drivers should wear masks. So I think if everybody wore a mask, you had all the windows open, even if it's chilly, I think it'd be good to open all the windows. Then I think you're going to protect yourself as much as you can. So that would be, I think that'd be a practical solution to that sort of scenario.
Yeah, do you have anything else to add, or should I go on to the next one?
I think Ed summed it up.
Well, Ed, you brought up at the end of that description in terms of opening the windows, that's actually an engineering control, right, in terms of ventilation. And so we've gotten a couple of questions and comments about sort of closed spaces, ventilation. Is there anything that the two of you could help us out with in responding to questions about these, in a clinic setting where you're in a closed space, and different types of engineering controls that are available?
I can respond in general to that. That I think is worthwhile to think about the biology of this virus, as well as rhinoviruses, noroviruses, and other nasty viruses that cause illness. that are transmitted in the air are highly susceptible, and we've seen it at least preliminarily with this particular coronavirus, to UV. And when I say UV, sunlight, just regular old sunlight, that will essentially rip apart these viruses. This one has an envelope, so it's a kind of a fatty layer on top of that encases this virus on a very, very microscopic scale. And UV kind of defats that. It kind of rips it apart to where it causes it not to have the ability to replicate in cells, because viruses are neither alive nor dead. They only use our cells to replicate themselves. And so if we reduce the infectivity by destroying its ability to replicate, then it can't get us sick. So when we stay out of enclosed areas or ventilate, as Ed had mentioned, in something like an enclosed space, like a bus or that kind of thing, and get it into light or other methods like circulation, increased circulation, then it'll further reduce the possibility of us coming in contact with the multiplicity of viruses that it probably takes to infect us. So those types of controls that would be considered, I guess, Amy, engineering controls that one can do by opening a window and and getting out in the sunlight. Those are practical solutions that we all can do. And we have a decent idea right now that this virus is not, Superman. It's not indefeatable. And with that, and it is susceptible to those things. So I would think that it doesn't take anything too terribly fancy to do that. Respiratory protection, of course, is also an added benefit. If one finds themselves in an enclosed space, that they will have a high probability of coming into contact with individuals that are infected and are infectious. And just a little bit about, I know we only have a couple of minutes, but about what we're now understanding about the pathophysiology of this disease and how it relates to transmission. And remember, this has not been studied for years and years. So I know there's a lot of frustration, like, well, why aren't the public health officials telling us this stuff? Or, it seems like they're rejecting this information. That's not it at all. They're trying to put together what's coming literally day by day as we try to respond to this. And everybody's in the same boat here. There's not like people are holding on to data or otherwise that would inform in such a way that we have this all wrapped up, because we don't. And I can say that with confidence because I'm one of the responders. And so, trying to understand about pathophysiology of disease and how long this stays around. What we know right now, and this is coming out in the literature, is that this hangs around a long time. And it's not a disease of chronicity. I don't think we're ready to go down that road yet. But it's not here and gone transient like one would consider a 2-day or 1-day cold, to where you're non-infectious. That's just not what's happening right now biologically. And so that has repercussions on the potential for transmission from infected individuals, and especially with what Ed had mentioned earlier, which is this asymptomatic, this what we're considering kind of this prototypical healthy young man or woman that is completely asymptomatic but may be shedding virus, and may impact high-risk populations. It looks like this virus is something that hangs around for an unusual amount of time, and unfortunately in those mucosal spaces that viral shedding can take place, your nasal carriage and things like that. So as this develops, and as we learn more about this, it can really inform about transmission, all the things that we talked about today in terms of mask use and engineering controls to protect yourself against the virus.
Great. Well, thank you both so much. I know that there was a ton of questions, and also hats off to my occupational health and safety colleagues. There's a lot of policy questions that came up, and we're not ignoring them. We're just trying to focus on this bit of the conversation. And I encourage you in your evaluation to add different types of Q&As, different kinds of discussions like this, so that we can carry on the discussion. We learned a lot of really important information about airborne and about the virus being airborne and its infectiousness. And I think that we can apply a lot of what we learned today in some of our work, and yet there's still a lot more that we need to do in terms of making sure that those that don't have control over their workspaces, who need to be in the workplace, who are in some ways are you know, have to go there to survive, that we need to figure out what can be done to make sure that they are safe. So there's a lot more to this conversation, and I really just want to thank Dr. Chad Roy, Dr. Ed Zuroweste, thank you so much. An evaluation is coming your way. Please give us suggestions for additional pieces. We'll get a recording out to you. We'll get a resource list out to you. We will then take a moment and go through all of the questions that you asked, some of the resources that you provided, and we'll follow up with this. And again, it's an ongoing conversation. Migrant Clinicians Network isn't going anywhere. We're here to try to respond to them and to bring in experts like Chad Roy and be able to sort of figure out sort of what that means for all of our clinicians who are on the front lines of this COVID-19. So with that, I'd like to say thank you again. Thank you to all of our participants. And please fill out the evaluation. It's a CME CE event. You have to fill out your evaluation if you want those credits. And then, but more importantly, we really want to know the topics and how we did and what we can do better. So please take a moment and take some time with that evaluation. All right, I think Martha, you can close it off.